the title of my presentation today is about an ameliorant, very important for ameliorant for soils, biochar. First, I would like to say a few words about soils, soil preservation and keeping soil degradation, at very low levels is a priority for the European Union, especially in the new law for soil monitoring, called the so-called Directive of Soil Monitoring and Resilience, which was approved by the European Parliament in October last year. We are a team of scientists who study the biochar, as I said, a promising soil ameliorant, a biochar influence on degraded soils, on polluted soils, and how it influences soil properties and soil characteristics. A few words about the heavy metals and what their behavior in soil is. They can cause deleterious effects on plant health and eventually on human health. Their behavior in soils depends on soil pH, soil cation exchange capacity, organic matter content. When they are absorbed by plants, they cause the formation of the so-called free radicals or reactive oxygen species, which cause oxidative stress and can eventually change the metabolism of different compounds like proteins, nucleic acids, lipids, etc. The actor of our presentation today is the element copper. It can be introduced into the environment and in soils by anthropogenic activities like industrial processes, addition of fertilizers and pesticides, waste disposal, mining, fossil fuel combustion. This presentation deals with copper pollution of soils introduced by activities. This slide can illustrate the structure of biochar, this promising soil ameliorant that can absorb heavy metals and also affect climate change, like gases, and influencing emissions of CO2 gases and other gases from soils. You see here the scanning electron microscope picture of the meso, micro, and nanopores of biochar. It has a hydrophobic core and hydrophilic shell. Here, I don't have a pointer to show you. But the hydrophilicity of biochar is caused by the presence of polar groups, hydroxyl, carboxylic, amino, etc. Its structure can be modified by the addition of acids and alkalis and salts, other polymers, which can make the biochar more reactable to different pollutants, organic or inorganic. This is the biochar that we used in our studies. It was produced by pyrolysis of birch, acer, ash, and maple. Pyrolysis, if you have heard, is a process of combusting organic materials in a medium lacking oxygen. So this slide is very important for soil scientists and for geologists. It illustrates the most important physical-chemical characteristics of biochar. The biochar normally has slightly alkaline pH. This biochar that we use has cation exchange capacity. It means it can absorb and desorb and exchange cations. So it has some hydrophilic carboxyl and phenolic and hydroxyl groups, so that it possesses CEC, cation exchange capacity. The organic carbon content is okay, determined by wet combustion method, potassium dichromate. It has 20% carbon, but at 1000 degrees, dry combustion, when it is combusted, the composition of carbon is 64%. The biochar that we introduce is not polluted with heavy metals, so that we should be assured that we are not introducing metals in soil when we try to ameliorate eventually soil pollution. So it has soluble elements which are below the norms according to the Directive 1999 of the European Union. The total metal contents of biochar are also below the levels of the International Biochar Initiative. This slide illustrates the structure of biochar and what kind of organic chemical groups it possesses. This is CP mass, cross-polarization magic angle spinning, nuclear magnetic resonance method, which determines different kind of groups, carbon groups in biochar. So the biochar that we use possesses more than 50% aromatic carbon, waste carbons, which are part of lignin structures, and also olefinic carbons. So this is a map that illustrates the sites that we sampled. They are from the metal copper producing facilities. Open Astarer Medet is the open pit copper enrichment plant, the biggest in southeastern Europe. So here we analyzed samples from the industrial side of the complex, from the tailings, which possess waste materials after copper has been extracted. These two here, I don't know if my pointer is seen. So the other copper smelting facility is Aurubis, the world-famous German group for smelting copper. So the copper concentrate from Astarer Medet has been transferred to Aurubis, appeared up here. Here is Aurubis, this is Aurubis, this is Astarer Medet. So we sampled here. Then here is the Medet complex. Also a mine where we analyzed reclaimed land, waste tailings and substrates that were reclaimed that passed biological reclamation. So, but still all these samples contain elevated contents of copper. Here Aurubis build-up site. Okay, the pointer is not seen. So we were not allowed to enter inside the facility. So if you see the numbers, we sampled from here, yes, we sampled from numbers 1, 4, 5, 6, 8. These were soils, alluvial, diluvial soils, polluted with metals coming mainly from aerosol contamination, aerosol pollution from the fumes which are generated after copper smelting. This is the other site mentioned, Astarer mine, a mine here, and this is the site where we sampled. You can see these bullet numbers where we sampled samples from the tailings, from the waste substrates after copper was extracted with sulfuric acid. And the last site is close to the Medet mine from the Astarer Medet open pit facility. So here we sampled soils from reclaimed materials, which had undergone biological reclamation in the year 2006. So more than 20 years of biological reclamation, but still copper was present in soils. So this is a very important table for soil scientists and physical chemists, but I will try to summarize, not be so specific. After we add biochar to the soil at 20 weight percentages to all these acidic soils, we get we achieve raised pH. From 4.6, it raises to 5.4 or 5.7. Then the total organic carbon also is increased, which is a good result. electrical conductivity is decreased, and redox potential, especially at the tailings from negative prevailing anaerobic conditions to oxidative. So 100, nearly 200 millivolts, which is a good effect from addition of biochar. Cation exchange capacity is also raised, not so strongly as the other parameters are improved, but still, because biochar does not introduce a lot of cation exchange sites, but still it is higher than the control variance. Here you can see our greenhouse experiment. So we mixed biochar with the soils or the substrates from the tailings. We introduced these samplers where we could extract soil solutions. You see here. These bigger pots are with the substrates. You see this rocky mass, and now biochar is added here. Again, a photo from our greenhouse experiment. So this slide possesses different graphs, but I will just generalize that after addition of the biochar, we get decrease in the solution of copper and almost all heavy metals like zinc, also lead, on manganese here in some sites. This is with the letter C indicates control, the control variant. Here on this part of the slide, you see these high bars with a very high concentration of copper, iron, zinc, manganese, and then after addition of biochar, the heavy metals and especially copper are totally fixed, becoming unavailable to plants and to leading to a decreased concentration in soil solution. Only for some unheavy metals like arsenic do we observe increase at some points after biochar addition, because it leads to increase of soil pH, so hydroxyl ions desorb the adsorbed arsenic, which was the reason for the increase of chromium. And for the cationic heavy metals, we get quite a strong fixation in soils and eventually decrease the concentration in soil solution. The biomass of alpha plants, I didn't say that we planted the biomass, the yield is increased following biochar addition in the samples from the 3 sites. This is the heavy metal content biomass. Again, we have a decrease of copper, manganese, chromium, arsenic in the biochar treated variants. Only in some variants, like for manganese here, we get not so strong decrease in the plant biomass. Graphs show that biochar can decrease the bioavailable form of heavy metals into plants, which is mainly represented by the free cationic species. This table, I will not be very detailed, illustrates the simple and multiple regression models of selected metals in plants and the different extractants. I forgot to tell you that we extracted metals, extractants, water, 0.01 molar calcium chloride and ammonium nitrate. The last 2 extractants are standard solutions for predicting plant bioavailability. And here you see that for the elements copper, zinc, manganese, lead, and arsenic, especially for the extractant calcium chloride, we get a very good predictability for their extraction ability and a very good predictability for the bioavailable forms of heavy metals. In addition, we carried out geochemical modeling by using a geochemical program, Visual Mintech, which can differentiate between the species in the soil solution, free species, copper ions, also complex species. And this table here, huge table with numbers, so I can just draw your attention about copper free ions, which are the most deleterious, the most bioavailable to plants when they're at high levels. They decrease after biochar. This tendency is followed also for the other cationic metals and or even for arsenic and chromium. This is the other side with the reclaimed soil. Again, we get here nearly 15 times less a decrease of the free most available metal ions to plants following addition of biochar. Biochar, in addition, leads to complexation of heavy metals, and especially copper, with organic ligands in soil solution, maybe mostly fulvic acids here, because they form the strongest complexes in water solutions with the heavy metals and copper. This table is interesting because it illustrates the speciation of metals in soil solution for the tailings, mine tailings, which are not soil actually. But it is a miracle because these toxic substrates can be turned into soil with live microbes in it after biochar has been added. Here, the tendency is different as from the acidic soils. For copper ions, we get initially in the controls the highest dominance of sulfate species, copper sulfate complexes, which are very much decreased after biochar addition, and free ions are available. One might say that free ions are very mobile. They can be toxic to plants, yes. However, here we have in the substrates general improvement of physical-chemical properties, of pH, of cation exchange capacity, of electrical conductivity, of redox potential, and all these positive outcomes influence the germination of plants, even in these toxic substrates. These are the conclusions. Most of them I always mention to you. But the most important thing is that biochar can lead to yield formation, even in toxic substrates. It decreases 4 to 15 to 13 times it can increase the biomass and can decrease the concentration of toxic metallic ions in the plant tissue. However, our long-term goal on this project, financed by the National Science Fund of the Ministry of Education of Bulgaria, is to monitor biochar long-term stability and its reliability in fixation of metal pollutants in soil. That's my presentation. Thank you. If you have questions, I would like to answer. Thank you.